So the cell membrane, really important, deserves its own video. So two functions. It gives the mechanical strength and shape to the cell. And secondly, its selectively permeable nature lets things in and out of the cell. So composition, it's made of four things as I mentioned last video. The phospholipids, which we might need a reminder on. The head is the phosphate group, the negatively charged, which means it's hydrophilic. You can remember because water is polar and it has that negative charge. The the fatty acids are neutral, non-polar, which means they are hydrophobic. And the inclusion of these two things, so we don't need to know how, but they allow things to be selective permeable. The next part is proteins. There are 5 different kinds of proteins, and I think it means the integral proteins, etc. There's the channel proteins. Carbohydrates, the little dots coming off from the cell, are cell markers. They're ID tags or antigens. There are two types. There are glycoproteins and there are glycolipids. Glyco for carbohydrate, to do with sugar, and protein and lipid are the two remaining, cell parts. So there are cholesterols in animal cell membranes only, so not plants. They lend stability to the lipid bilayer, and they prevent a drastic decrease in fluidity at low temperatures. That's what cholesterols are really important in the structure. So the cell membrane model looks like this, as opposed to the older Davson-Danieli membrane model, which actually looks like, I'm gonna look it up for you. And Davson-Danieli looks like this, where they were unaware that the proteins were embedded. So the current one is a Singer-Nicholson, so don't get surprised if you see that. Davson-Danieli, from the electron micrograph, this is all they could see. They thought that the proteins were sandwiched and create a layer like this, but in reality it is the proteins that are surrounded by phospholipids and integrated in and throughout. So this is an integral protein, and these are peripheral proteins, the ones where they're sideways. The outside is hydrophilic, you can see from the heads, and the inside will be hydrophobic. And this kind of like balance allows for it to be selectively permeable. It's called the fluid mosaic model because the phospholipid tails form an oily layer, which you can see in the micrograph. And the mosaic because there's so much stuff, so many colorful things embedded within. The cell membrane permeability. So for things that are impermeable, there are macromolecules. Things that are too big won't fit through the hole. Charged particles, things that, I don't know, have jail sentence. They're charged. They can't fit through. Without help, at least. And small uncharged lipid-soluble molecules can fit through, such as gas, alcohol, and waste. This is a really good thing that they can fit through. Otherwise, it'd be a big issue. So, and water, as a result of water's partial polarity, it is not lipid-soluble. So water has difficulty crossing the cell membrane. However, water is permeable through specialized pores called aquaporins, which is why it's allowed to pass through for osmosis. So, transport across the membrane can either happen passively, without ATP, or actively, and that would require ATP. So there are 3 general means of transport. There's diffusion, transport by proteins, endocytosis, and exocytosis. But this is a different kind of thing we'll come back to later. First of all, diffusion, considered passive, which means without ATP, is the net movement from a high concentration to low concentration, meaning like rolling down a mountain. You move from the top to the bottom, and you're moving down the concentration gradient. Osmosis is this specialized kind of diffusion for water only. So notice here, there's a lot more solute. There's more salt on this side, and there's a lot of water on this side. So the water is the only thing that can pass, right? So through the aquaporins, so the water will make it through to side B. And the net movement creates pressure as it moves through, called osmotic pressure. So eventually, it will even out, and there will be an even amount of water between these two, but the amount of solute will remain. You can think of it as high solute means less water, less solute means more water. And the water is the thing you're trying to get to move. Tonicity is used to describe the situation. There's isotonic, hypertonic, hypotonic. Isotonic means from the cell inside and the outside the cell, it's the same. Hypertonic means that there's a greater solute concentration inside Oh no, sorry. The cell is in a hypertonic solution, which means there's more solute outside. And what this means is the water from inside the cell leaves to go out because there's more water inside than there is outside. Hypotonic is the opposite. So when a lot of water enters the cell rapidly, because the solute concentration outside is less than inside, the cell could explode because it will swell up and might even lyse, or this term is lysis. So crenating is where the cell shrivels up because it's losing too much water too fast. In plant cells, it's surrounded by a rigid box-like layers of cellulosic cell wall. So in plant cells, they have their own terms. When they're placed in a hypertonic solution, only the large central vacuole loses water, and the cell membrane pulls away from the cell wall. So this is called plasmolysis. So just add the word plasmo in front of lysis. Plasmo for like plasma membrane. And this is like ecrenation, but the cell will hold structure because of its rigid cell wall and large central vacuole. When plant cells are placed in a hypotonic solution, cytoplasm and large central vacuole will gain a lot of water, and then it'll swell up and push against the cell wall. But I don't think it will. It usually won't lyse because it will just be turgid against the cell wall, pressing against. So turgor pressure and osmotic pressure are equal, and it's just that plant cells is turgor pressure. So this is the turgid plant cell. Maintains an erect position. Remember that it's erect. In single cell organisms, such as protists contain contractile vacuoles, which I'm pretty sure means they can stretch and close in. When they're placed in a hypotonic solution, water flows in and excess water collects into these contractile vacuoles, which expel and pump water out to remain safe, and you would need ATP for that. And the opposite for hypertonic, and the vacuole becomes less active. For animal cells, the most important one for us, homeostatic mechanisms maintain the water content, and body fluids controlled so they're always isotonic to cells. So if you think about it, your blood cells aren't suddenly going to explode because there's too much water or too little water. So when we have too much water, what do? We pee, we sweat, and we exhale, and there's some moisture from our breath, especially for mouth breathers. When we have insufficient water, well, go drink more water, go pee less, right? So animal cells are usually not exposed to hypo or hypertonic solutions, but plants can be. For example, when we did that potato lab, or whatever vegetable or fruit you brought in, and you would need to place them in water and watch them lyse or shrink. So transport by proteins, also known as facilitated diffusion for one, is passive. Facilitated means that they're watching, but they're not helping you. They're passively helping you. If you remember, I said that charged ions are like, I don't know, they have a jail sentence. So they have a charge and they can't leave. But they can leave, they can go on a little break, as long as they're facilitated. So this would happen through protein channels, which allow certain things to go down the concentration gradient. So they still can't run up the hill. They've sat around in jail for too long. So they can't go down the they can't resist the concentration gradient. So additionally, glucose and amino acids are also carried through with carrier proteins. These still go down from high to low, but instead the carrier proteins aren't just an open channel. They bind to the substance and change shape, like so. Like this diamond-shaped glucose or amino acid, they would bind through it and let through. So only glucose and amino acids for these. Only glucose and amino acids, really important. These two biological molecules. Active transport, instead, maybe they paid bail, and now there's a prison guard exporting them out, and they can finally go up the hill they've wanted to go up for this whole time. So it can go up the concentration gradient from a low concentration to high concentration, which means energy is needed. You need energy to power that bus you're taking out of jail. So ATP is required to cause the carrier protein to combine with the molecules to be transported. Mitochondria are in abundance where this happens, such as the sodium and potassium pumps in your brain, your nervous system. You need to keep the sodium out while accumulating potassium in. So this is what the three-step process looks like. The carrier protein will allow a certain amount of sodium in for the sodium potassium cell, and ATP will be transferred. Adenosine triphosphate will be turned into diphosphate as a phosphate group is used up for its high energy bond to kind of power this carrier protein. So the Na is pumped out used from this kind of valve, and potassium can come in as the carrier protein changes shape to adapt to it. And then finally the potassium is let out. So endo and exocytosis require ATP, but we don't need to know how the ATP is used here. Endocytosis, or cell, endo meaning in, means we take something in. There's phagocytosis and pinocytosis. Phagocytosis, like macrophage, like cell eating. Think of your cell turning into like a Pac-Man. It'll eat something, like a white blood cell will eat bacteria, and then trap it in a vesicle to destroy, like with enzymes. Pinocytosis, or cell drinking, is for smaller solutions, like liquids. Exocytosis is the opposite, so we don't have to differentiate between eating and drinking. You'll just eject and excrete them when your waste proteins, for example, are trapped in a vacuole, they fuse with the membrane and are let out. Factors affecting the rate of transport in cells. When there's a great concentration difference, like if you think about it, the steeper your hill is, then you're going to roll down that hill really fast. But if it's a nice, gentle change, like 50% and 40%, there'll be gentle movement. For the size and shape of molecules, smaller and smooth molecules move across faster than the large and varied shape ones, such as those in like complexes. Size of pores allow more molecules to pass through faster than small ones. Well, obviously, if the hole is bigger, then more can pass through quicker. And temperature, according to kinetic molecular theory, there'll be more things moving in and out because they'll be hitting the right spots correctly at a faster speed. Oh, and this is something additional about the cell membrane. The surface area of the cell membrane is a square function, whereas the volume is a cubed function. Makes sense? And if you think about it, the cell will outgrow, the volume will quickly outgrow the surface area, if you think about a sphere or a cube, which means that it's too small and the diffusion of gases, waste, and such becomes too slow to keep up with the volume. To overcome this, you can divide the cell by mitosis, or the cell could have extreme folding like in the intestine. Yeah, extreme folding like the villi, to overcome this and have a high surface area as well as a high volume. And that's it.